Okay, so we're going to switch gears a little bit, talk about genome analysis, another area that I'm quite excited about. I'm going to present this on behalf of my postdoc, Hayu Mao. Essentially, we're looking at a lot of different ways of extracting genome analysis. So I'll motivate the problem first. Clearly, genome analysis is important. If nothing else, COVID-19 pandemic showed that to us, right? Because the sequences that I'm going to show you enabled a lot of development of vaccines and variant detection, etc. And there's a lot more. So that's just one example. It enables understanding of our past genetic variations. It enables developing personalized medicine, disease outbreaks, and who knows what else, many applications. I've already said the first one over here. So if you look at modern genome sequencing machines, they look like this. This is one small one. This is the nanopore technology. And it's a widely used sequencing technology. Like other techniques, they extract small randomized fragments of original DNA sequence. It's called reads. And then you need to basically put this thing together to understand what the genome looks like. But I'm going to focus especially on this ONT sequencing device because we've been working on trying to make genome analysis more embedded and portable. And there are a lot of restrictions over here, clearly. But this is a portable sequencing device. It's high throughput. It's cheap. You can get it for $1,000 or so. But there are other genome sequences that also exist. This is just one example, a set of examples. They all produce data with different properties, and they're more high throughput sequencers that have been developed. This is the nanopore sequencing technology that we're going to focus more on because it generates a lot of data very, very quickly. And this is actually, a lot of these were shipped to all over the world to sequence the COVID-19 RNA, and they're still being used for that. Now, why is this why is this very interesting as a data-intensive application? Because the hardware technologies that we built to sequence genomes have been extremely successful. They're not perfect, but the cost of genome sequencing has been reducing significantly. So if you look at this graph, this is cost per human genome. And for reference, we have Moore's law curve. You can see that with the introduction of high throughput sequencing technologies, the cost has been reducing at a much faster exponent. This is the introduction of nanopore sequencing technology. So the cost of genome sequencing is reducing much faster than the cost of a transistor, which means that we have a bottleneck that kind of looks like this. We have a machine that's very specialized that can generate a lot of data at a much lower cost than a machine that's general purpose and that's governed by Moore's law, let's say. So the amount of transistors that we can put on this thing on the right is not going to keep up with the amount of data that's going to be generated over here. So that's one of the issues. This machine on the left is very specialized. This machine on the right is general purpose. And then we need to move the data from the machine on the left to the machine on the right. So that causes a lot of data movement. I'm going to quantify that for a very simple analysis soon. So I think we have this problem in genome analysis much more than in other domains. But if you look at this picture, replace that Illumina sequencing machine with a video camera. Video camera essentially has a similar property. It's a very specialized device. Replace it with web clicks, etc. So basically, this is why we're having problems in our systems today: data movement and non-specialized computation capability. So if you're interested, we have a lot of information on this. Before I go into a little bit more detail on this topic, you can find actually our genomics course as well as processing in memory course. Now, let me go into a particular example of how we're accelerating this. This is a big area. I'm going to give you an example which we published in Micro last year. So basically, you have multiple steps in genome analysis. I didn't give you the full picture, but once you actually get these reads, you need to reconstruct them. After you reconstruct them, you need to analyze them. And then after that, you need to start asking questions. Oh, is this the gene that I'm concerned about? Or how does this genome look like from compared to many other different genomes I have in my database, right? So in all of these analyses, actually, you have large data movement between multiple steps and a lot of wasted computation. And in this work, we're going to target both of them. Why do you have a lot of wasted computation? Because you need to do some computation to discover what's the important part to focus on in the genome, for example. If you don't do that computation, you cannot actually decide which part to focus on. So our goal will be to actually eliminate that wasted computation as much as possible to begin with. So let me give you an overview. So the GenPIP is a fast and energy efficient in-memory acceleration system for the genome analysis pipeline. And it tightly integrates multiple genome analysis steps. It has two key techniques that we build on. I'll not go into a whole lot of detail because there's a lot of technicality over here. One is basically chunk-based pipeline. As opposed to working on large chunks, large pieces like reads, work on smaller pieces, smaller chunks. I'll give you this pictorially soon. And then the other technique is early rejection. The idea is to stop or timely stop the execution on useless data by predicting which reads, which part of the genomic reads will not be useful. And we show that it can outperform state-of-the-art software and hardware solutions using CPUs, GPUs significantly, and also optimistic processing in memory systems significantly. I'll mention what we mean by optimistic PIM. Okay, let me give you some data here. So you sequence genomes. This is the nanopore. If you look at a nanopore device, you have a nanoscale hole, and you pass the DNA strand through that nanoscale hole. As the DNA strand passes through this nanoscale hole, the nanopore device measures the current. And different amounts of current corresponds to different bases. It's not a perfect process. It's very error prone. So you need to actually, what happens is you take the data out of that device, these signals, and store them in storage. And then you need to do what's called base calling. Base calling basically converts those signals into text. Is it ACGT? So that's the question we ask. And usually you use neural networks, sophisticated neural networks today. There's a neural network, and then you get the ACTGs. And you can see that this is a lot of storage accesses. And then now you construct the read, and then you put it into storage again. And then you take it out of the storage, and then you do something called read quality control. How good quality are these reads? Can I actually proceed with computation? You eliminate some of them. Okay, you move the high quality ones to more sophisticated processing. Now this is called read mapping. Basically, you take these reads and map it to a reference genome so that you can actually try to reconstruct what you've sequenced. For example, you have sequenced COVID-19, which is a small RNA. You can compare it to many, many other genomes, many other virus RNAs, and then you can see how it's evolved potentially, right? But you need to be able to do that, you need to do read mapping first. And then you do the read mapping. Some reads will not map because of errors in the machine, because of variations in the genomes, etc. Then you need to store the remaining parts. And then there's more analysis on top of this for high-level questions. So you can see that storage is involved in many places, right? We want to eliminate those storage by integrating things as much as possible. So let me give you an example of the amount of data. So raw signals in a single human genome is about 4 terabytes over here, as you can see. You do base calling reduces it to 500 gigabytes or so. You do read quality control, you eliminate some of them. And then you do read mapping, it goes down to 400 gigabytes. This is a single genome. It's not, if you actually are doing metagenomic analysis, you may be working on thousands of these together. So it's actually a lot of storage in the end. So you can see that there's large data moment between these different genome analysis steps that we're trying to eliminate. And then there's wasted computation. So here you get all of the reads. And then after read quality control eliminates 20%, let's say, and then read mapping eliminates 10 more percent in this particular data set. In some cases, it's actually much more. So we actually do computation. We actually do base calling on all of them, even though some of them are going to be eliminated. And base calling is actually very costly computation. People actually use neural networks, as I mentioned. That's another area that we're working on. So basically, because of low quality reads as well as unmapped reads, there's a lot of computational useless data. So what do state-of-the-art works do? Actually, there are a lot of state-of-the-art works. I think like most state-of-the-art ones use processing in memory using non-volatile memory technologies. For example, there's this work that does dot product operations in RRAM type of structures, which I think fascinating to do base calling acceleration. And then there's another work that does search and addition to do the read mapping, again, in RRAM type of structures. So they reduce the data moment in a single genome analysis step. Processing in memory enables that. But they exacerbate the data moment overhead between analysis steps, because if you reduce the data moment in a single step, the other parts of the data moment become the bottleneck. And essentially, no prior work tackles the data moment between the analysis steps and also reduces useless computation. That's what we wanted to do in this work. I think there's more to do in this area. So our goal was to efficiently accelerate the entire genome analysis pipeline while minimizing data moment and useless computation. Let's take a look at the potential study first. First, we quantify the potential benefits. Results are normalized to the performance of a good GPU, essentially. MVM-based PIM accelerators are actually pretty good compared to a GPU, 2.7x, as you can see. Now, if you eliminate the data moment between these two different accelerators so that they communicate with each other nicely, you go up to 6.1x, which is nice. And then if you actually eliminate the useless reads in an ideal way, you go up to 9x. Again, this is a single data set. In other data sets, your mileage may vary, let's say. But 9x is not bad. Our goal is to actually get that 9x. So, okay, now we're going to go into a little bit more detail. As I said, there's chunk-based pipeline. This enables fine-grained pipelining of genome analysis steps. You process reads at a chunk granularity, as a small granularity, like a subsequence granularity, like 300 bases as opposed to 1 million bases. In nanopore sequencing, you get 1 million bases. Okay, so what is this benefit of this? This increases parallelism by overlapping the execution of different steps. And it also reduces intermediate data by computing on data as soon as it's generated. You don't wait for 1,000,000 bases, you wait for only 300 bases, and then you do some computation. And then this is also going to enable us to do something else, as we will see, which is this basically. It enables opportunities for early rejection. Because if you actually analyze a small portion of the read, and if you say, okay, that's not going to be good quality, in my opinion, based on some good metrics, hopefully, you don't need to base call the remaining part of the read. So let's take a look at this pictorial. This is our conventional pipeline. You base call the entire read, and then you do some assembly on top of this, and then you do read mapping. Instead of that, we do base calling of a chunk, and then we start quality control and read mapping. And then we pipeline the quality control and read mapping of the first chunk with the base calling of the second chunk. So this way you can save cycles. So the basic idea is essentially pipelining fine grain, but this also enables us, as I said, for example, if you discover over here that this read is not going to be good quality, you can stop the base calling of the remaining chunks. Okay, so early rejection, I kind of giving you the idea. You stop the execution on useless reads as early as possible by using a small number of chunks to predict the usefulness of a read. I already said this. So you base call a small number of chunks, you check the average quality. If it fails, you stop the analysis. If it passes, then you base call more chunks. And then you map the base call chunks to a reference genome. And then you check the mapping scores. This is already done in existing genome analysis steps. If it fails, then you stop the analysis again. And nanopore sequencing device is nice because it enables you to stop base calling. It has this nice mode that we are taking advantage of right now. If it passes, then you base call the remaining chunks, and then you execute the remaining parts and read mapping. Yeah, basically there are two places where we can do early rejection. We can predict low quality reads using chunk quality scores over here. And we can predict unmapped reads using chunk mapping scores over here. Okay, so this idea actually is more general than processing in memory. It can be applied on CPUs, GPUs, and processing in memory. But we use processing in memory because, as we've been talking about, it's really the state of the art in terms of performance and energy. Yeah, and we also apply these techniques on CPU and GPU baselines to observe speed up energy savings. I will not go into a whole lot of implementation since we don't have a whole lot of time, but you can read the paper for more detail. Essentially, we have a GenPIP controller that gets raw signals, stores them in embedded DRAM, and then it sends a chunk to an in-memory base caller, looks at the quality scores, and then if the quality score is good, it continues with computation. And then continued computation goes to an in-memory read mapping engine. As I said, these two are based on prior works using RAM, but it has some R design as well. Okay, as I said, I'm not going to go into a lot of detail. There's a lot of detail over here, but you can do early rejection at different parts of this. So we envision this as a single system without moving the data. And ideally, this should be integrated into the sequencer also. Okay, so let me give you some results. This is all, yeah. Oh, 5 minutes. That's more than what I need, I think. I don't intend to talk a lot about results over here, but I'm happy to take questions. So we actually evaluate this rigorously with both Verilog components and simulators. And there's more on our methodology in the paper. We have CPU, GPU baselines, and optimistic integration of the PIM accelerators. Optimistic means you don't have data movement between those two PIM accelerators. This doesn't happen today. And we use two different data sets, as you can see, a small data set and human data sets. And if you look at the results, these are the summary results. There's more detailed results in the paper. Compared to a CPU, we get more than 41x performance improvement. Compared to a GPU, it's more than 8x performance improvement. And compared to this very optimistic processing in memory, we get 40%, which is not bad because this is very optimistic, which is not implementable today. And both chunk-based pipeline and early rejection are actually important for speed up. If you look at energy, which is quite important also, we get significant energy savings as well. So you can see that it's 30x, 20x, and about 40% compared to the PIM. And in this case, early rejection is especially critical for energy efficiency because it gets rid of useless computation. Okay, there's a lot more in the paper. I invite you to take a look at it. I'd be happy to talk about this more, but I'll try to end early. Okay, this is a summary. I don't want to repeat the same thing again. I'll just mention that there's a lot more work in this area. I think this is a fascinating area. There's a lot of work that's going on in machine learning today. I think machine learning is quite interesting, but the workload itself, machine learning, in my opinion, is actually relatively simple compared to the workload that we're facing over here in genome analysis. And genomics is just one part of the omics. There's also protein analysis, which is even harder, in my opinion. Okay, that's the paper, and you can find the slides and videos online as well. And we actually cover a lot of these topics in our computer architecture course and genomics course and PIM course. If you're interested, you can attend. And Juan and I have been organizing the real processing in memory tutorials at ISCA, ASPLOS, and HPCA. There's one we're organizing at Micro. I didn't have time to prepare the slides, but these are all available online. You can attend, ask questions, or even participate if you're interested. The Micro one will be completely virtual. Questions? That's a great question. Basically, we don't use a single chunk. We use a sampling of multiple chunks. But yes, if 300 by itself is too short.
So these chunks are based on a single large molecule from a nanopore.
Exactly.
This is not what we do for all.
So in so that's correct. So in nanopore gives you, let's say, 1 million bases. It's bigger than what you get with Illumina, for example. And we chop it up into smaller pieces, non-overlapping. Yes, that's correct. Yes. So in the end, you can get potentially some errors, but the amplification that you also need to do with nanopore, because you're error prone, handles a lot of those.
Part of the rejection.
That's correct. Yes.
You do try to compensate for indel before you reject them.
So depends on where you reject. In the rejection that you do in the read mapping parts, based on the mapping part, you definitely account for indels. But the other rejection is really based on pure read quality control. That is already done. So we basically do the read quality control in a let's say finer grain manner.